Continuing now with the Alan Gewirtz Memorial Scholarship, Alan was a past president and very active member of the OTS for many years before his untimely death due to cancer. And this scholarship is awarded in his memory to scientists who epitomize his dedication to the application of basic research to clinical problems and solutions. And this year's winner is Karyn Schmidt, who is going to talk to us about inhibiting the androgen receptor interaction with the long non-coding RNA silencer using 2' FANA-Modified oligos to decrease melanoma invasion and proliferation. Karyn?
The green button to advance.
Okay, and how do you go back?
You can go back with the red one.
Okay, thank you. Good morning, everyone. Thank you so much for coming so early in the morning. And I'd especially like to thank the committee and the organizers for this incredible honor. I'm very excited to be attending my very first OTS meeting, and so far I've thoroughly enjoyed it, so thank you so much. So as Art said, I'm going to be telling you a little bit about my postdoctoral work investigating the mechanism of a lincRNA in cancer. But in the more global picture, I hope you can take away a little bit about how we might be able to target long non-coding RNAs therapeutically. Okay, so just a few key points that I'm going to be presenting today. So SLNCR is a novel long non-coding RNA that has a role in both melanoma invasion and proliferation. And it does this by binding to and regulating the activity of the androgen receptor, or AR. AR binds single-stranded RNA in a sequence-specific manner, and because of this, I was able to design single-stranded steric blocking oligos to inhibit the interaction of SLNCR and AR. And these oligos actually decrease melanoma invasion and proliferation. So first, what are long non-coding RNAs, or lincRNAs? In general, most of them resemble mRNAs in that they're spliced and polyadenylated. They can range in length from 200 base pairs all the way up to greater than 10 kb, and they can span very long genomic regions. And if we look at this pie chart shown here on the right, here we go. The human genome encodes, as we know, about 20,000 protein-coding genes. And the number of lincRNA species, and at this time, this was 2015, there were close to 60,000 different lincRNAs that had already been annotated. By some measures, this number is already closer to 100,000 lincRNAs. So the total number of lincRNAs greatly outnumbers the total number of protein-coding genes. However, their expression is exquisitely tissue and disease-specific. So at any time, you'll only see expression of a small portion of this total possible lincRNAs. Many of these have been implicated in cancer, as well as other human diseases. So because of these, they have interesting implications as therapeutic targets. However, because many of these lincRNAs don't have well-defined mechanisms, their therapeutic application so far has been fairly limited. As I said, the mechanism of many of these lincRNA species is as yet not well defined. And part of this is because the characterized lincRNAs so far have been shown to function through a wide range of possible mechanisms. So for example, some lincRNAs interact directly with protein cofactors to mediate their function. So for example, some lincRNAs interact with chromatin modifiers or transcription factors, and this can act to recruit them to specific genomic regions or sequester them away from where they normally function. Other lincRNAs can act as a scaffold, mediating formation of a higher order RNP complex. Other lincRNAs can interact directly with other nucleic acids to mediate functions such as acting as a microRNA sponge or interacting directly with mRNAs to regulate translation, splicing, or even elicit degradation of the associated mRNA. And I should also mention all of these functions are not necessarily mutually exclusive. So determining the mechanism of various lincRNAs can be quite the task. So when I joined Carl Novina's lab at Dana-Farber, I was interested in understanding the fundamental mechanisms of lincRNAs in melanoma, which is a malignant tumor of melanocytes. It's estimated that close to 90,000 new cases of melanoma will be diagnosed just in the US this year. And interestingly, there's a well-established gender bias in melanoma in that males account for 2/3 of all of these diagnoses. So although melanoma is not the most prevalent form of skin cancer, it accounts for over 3/4 of all deaths associated with skin cancer. And this is partially because melanoma is particularly lethal because we don't really understand mechanisms of melanocyte transformation very well. So early in my work, we used RNA sequencing to profile long non-coding RNAs expressed in patient-excised melanomas, and I identified a robustly expressed lincRNA called SLNCR, standing for SRA-like non-coding RNA. And as we can see in this qPCR, we confirmed that SLNCR is expressed in all melanomas that we checked, shown here. Well, we did not detect expression of this lincRNA in any normal melanocytes. So this lincRNA seems to be ubiquitously expressed in melanoma and is specific to the cancer. Moreover, we then interrogated the cancer genome atlas and found that higher expression of this lincRNA SLNCR is associated with shorter overall melanoma survival, and this is split based on median SLNCR expression. And we can see high SLNCR expression here in red, associated with shorter survival. Melanomas express 4 SLNCR isoforms that we've identified so far, shown schematically on the left. These isoforms are localized in both the nucleus and the cytoplasm. These are spliced and polyadenylated transcripts, and the most prevalent isoform is SLNCR1, and it's just over 2 kb long. So early on, to try to understand how SLNCR might be functioning in melanoma cells, I used RNA-seq to profile patient-derived melanomas depleted of SLNCR using 2 different siRNAs targeting the lincRNA. And as you can see in the heat map, we saw significant dysregulation in over 200 different transcripts. And remarkably, a lot of these genes are involved in hallmarks of cancer, such as cell adhesion and motility, cell differentiation, as well as cell proliferation. Consistently, if you knock down expression of the lincRNA using 2 different siRNAs, we see a significant decrease in melanoma proliferation of 2 patient-derived melanomas compared to scrambled siRNA control, indicating that SLNCR increases melanoma cell growth. Moreover, and this work was published last year in Cell Reports, I showed that the most prevalent isoform, SLNCR1, significantly increases melanoma invasion as well. These graphs are quantification from Matrigel invasion assays, and on the left is a patient-derived melanoma line, where if we knock down expression of SLNCR, we see a significant decrease in melanoma invasion. And conversely, on the right is a melanoma cell line which expresses lower levels of this lincRNA, and we can ectopically overexpress SLNCR1 and increase melanoma invasion, shown here. So through a lot of work I don't have time to go through today, I determined that SLNCR likely functions through interaction with various transcription factors. And as many of you know, RNA IPs are generally very low efficient, or have low efficiency, so it's difficult to work with the output from these, as well as the fact that transcription factors sometimes have very low expression in the cells. So as a workaround of these technical complications, I devised a novel approach called RADA, and this was published as a methods paper earlier this year. So just very briefly, what this method does is it couples an MS2-based RNA IP in which you use a flag-tagged MS2 protein to IP a MS2-tagged RNA from your cell. elute with flag peptide, and you can then take this eluate and use it as an input into a transcription factor activation array, which enables highly sensitive and relatively unbiased identification of transcription factors in this eluate. And using this methodology to identify transcription factors associated with SLNCR, I determined that SLNCR does indeed associate with multiple transcription factors, most notably the androgen receptor, or AR. And this is a nuclear receptor that's typically activated by binding of androgenic hormones such as testosterone. But interestingly, multiple labs had previously shown that lincRNAs can bind to and regulate the activity of AR. And I was particularly intrigued by this given the fact that there is a male-disfavoring melanoma gender bias, in which, again, men account for 2/3 of all total diagnoses. They also have a significant disadvantage in survival and time to first metastasis. And compared to females, men are 55% more likely to die from melanoma. And previous work has implicated AR in the melanoma gender bias, but molecular mechanisms underlying this really remain obscure. And moreover, what's very interesting is if we return to the Kaplan-Meier survival curve, where again, high SLNCR expression is associated with worse survival, and we then split this based on gender, we see that for both males and females, we do see the expected worse survival. But interestingly, if we look at low SLNCR expressing males, shown in blue, we see that they show worse survival compared to females at low SLNCR levels, shown in red. And this seems to correlate with expression levels of AR. So if we look at the low SLNCR melanomas, we see that males express significantly higher levels of AR, suggesting that high AR expression predicts sensitivity to this AR interacting lincRNA. So knowing all of this, I wanted to further interrogate the functional implications of SLNCR and the AR association. So first, I did confirm that AR is required for SLNCR-mediated melanoma proliferation and invasion. I then performed AR ChIP-seq from melanoma cells to identify where AR is binding in the genome. And importantly, I performed this from hormone-deprived cells. So my previous data indicated that SLNCR is regulating AR activity in the absence of normal androgen-induced AR function. And what's interesting is that if we compare the SLNCR-regulated genes that I previously identified using RNA-seq to the genes where AR is actively binding, we can see that AR binds to almost half of all of the SLNCR-regulated genes, indicating that AR is critically necessary for mediating this oncogenic lincRNA function. So I don't have time to go through all of this data, but over the course of my postdoc, I determined that SLNCR fine-tunes AR activity through two different models. And I use the word fine-tune because it's not a complete on or off switch of AR activity. Rather, it's kind of co-opting its activity to very specific regions in the genome. And it does this either by a cooperative binding model in which two transcription factors might not bind to low consensus DNA binding sites in the genome. However, in the presence of SLNCR, it can mediate formation of a higher order complex, bringing these two transcription factors that don't normally interact together, and together these transcription factors have higher affinity for these low consensus DNA binding sites. In another model, SLNCR can act as a tether, recruiting the androgen receptor to other SLNCR interacting transcription factors and recruiting AR to novel genomic binding sites. So knowing how critical the interaction between SLNCR and AR is, I decided to really dive into defining how this interaction is mediated. And consistent with results from Michael Rosenfeld's lab at UC San Diego, I found that the N-terminal region of AR is required for binding to RNA. And shown here on this gel shift assay, we can see that a probe corresponding to 28 nucleotides of SLNCR, and I determined this region through previous structure function analyses, specifically binds with relatively high affinity to the N-terminal region of AR. I then used a series of mutated probes to try to figure out exactly the sequence requirements for mediating the interaction to the androgen receptor. And what I found is that AR binds to pyrimidine-rich RNA sequences, specifically requiring the presence of at least one UCU, CCU, or A motif. And if we look here, the wild-type 28 sequence contains 2 such motifs. And if we mutate the one on the left, we still see AR binding. If we mutate the one on the right, again, we still see binding. However, if we mutate both of these motifs, we completely abolish the ability of AR to bind to this linker sequence. And to further confirm these results, I then shifted this probe upstream just slightly, so wild-type 17 sequence of linker that contains only one of these UC-UCC-A motifs. And to confirm that it does indeed require single-stranded RNA, if we introduce a few nucleotides upstream to induce formation of a stable stem loop, shown here, we completely abolish the ability of AR to bind to this sequence. And to highlight exactly how exquisitely sensitive AR is to sequence of the RNA, if we just swap 2 nucleotides within this motif, so going from a CU in the wild type to a UC in the mutant, we again can completely abolish AR binding to linker. Knowing this, I then designed single-stranded steric blocking oligos to inhibit this interaction, and these work either by binding to the androgen receptor called AR binders, or are the reverse complement to this linker sequence required for binding and are called reverse complements, generating double-stranded RNA that shouldn't be a substrate for AR binding. And as confirmed in this gel shift assay, introducing these sequences, and these are 2' FANA-Modified oligos, we can introduce these sequences to our gel shift assay, and as shown, any of these sequences very specifically blocks the interaction of AR and linker. I then tested if these oligos were able to negate linker-mediated effects, and as shown here, we did see that blocking the interaction of AR and linker inhibits linker-mediated invasion. So as shown before, ectopic expression of linker-1, again the most prevalent isoform, significantly increases melanoma invasion, and subsequent gymnotic delivery of these 2' modified FANA oligos negates linker-mediated invasion, shown here. In addition, gymnotic delivery of these oligos to patient-derived melanoma cells also decreases melanoma proliferation compared to a scrambled control shown in blue. So again, the key points of this talk that I hope everybody took away was that linker binds to and regulates the activity of the androgen receptor, and by really carefully defining the requirements for this interaction, I was able to inhibit specifically the AR-linker interaction to decrease melanoma invasion and proliferation. And just to leave you with a few perspectives about possible more global implications of this work, as has been a common theme throughout this meeting, I think we're learning a lot more about just how amazing RNA is and how many different roles it can play in the cell. And I think link RNAs are a prime example of RNA's role in fine-tuning gene expression. In addition, I think this is kind of a proof of concept work that you can inhibit the interaction of link RNAs and proteins as a novel therapeutic approach. I also want to mention that while my work focused on melanoma, linker and other AR-interacting link RNAs have been shown to play a role in multiple human cancers, including breast, lung, ovarian, there's a whole bunch of them. And finally, I just want to mention there might be a few advantages, specifically in this case for using a steric blocking approach in terms of link RNA therapies. So very few copies of link RNAs, especially linker in this case, are sufficient for oncogenic effects. So we want to make sure we completely block all link RNA activity. And specifically by binding to AR, we can even preemptively block lncRNA function even before it might be expressed in the melanoma cell. And lastly, this really enables inhibition of lncRNA-specific transcription factor activity. So rather than taking a sledgehammer approach in which we're trying to turn off AR function as a whole, which might have a lot of pleiotropic effects, using the steric blocking approach, we're able to just turn off the oncogenic effect of the androgen receptor. So finally, I'd like to acknowledge everyone who helped with this work, especially my postdoctoral advisor, Carl Novina, and our entire group at Dana-Farber. And thank you again for your attention. I hope I've told you a little bit about lncRNAs and possible therapeutic approaches. And thank you again to the organizers for this wonderful, wonderful honor. Thank you. I'm happy to take any questions.
And we may have time for one or two quick questions. Go ahead.
Hi, excellent talk. Quick question. Does it bind to other steroid receptors, like the glucocorticoid or the estrogen receptor? Because the N-terminal domains are quite similar across these receptors.
Yes. I don't believe it binds to the estrogen receptor or glucocorticoid receptor. I haven't thoroughly confirmed that, but my data doesn't suggest it. However, there is one paper suggesting that the estrogen receptor actually regulates expression of SLNCR. So that's something that we're following up on and might also have interesting implications for the gender bias as well.
I just have another quick question too. Do you know what upregulates SLNCR expression in tumors since it's not regulated in normal cells?
Yeah, specifically in melanoma, I'm not sure. The promoter does contain a CPG island, so it might be related to dysregulation of CPG islands in cancer. I can't say for sure right now. It could also be related to estrogen regulation.
Don't go away yet. I need to give you your presentation. Certificate here, so congratulations.
Thank you so much.